So the title that Björn and you probably gave me was to talk about the neuroscientifical perspective of early interventions. And of course, that's a huge title that could fill at least an entire morning. And I only have 20 or 25 minutes. So it's really trying to get from the initial steps of when people got interested on the little puzzle of the brain and how it gets connected. And one of the famous neuroanatomists, Ramon y Cajal, a neuroscientist, who very early on actually realized that the way a neuronal network is established is very much something that happens early in development. And so he clearly found out whatever the environment, the chemotropic environment of a neuron in its initial steps will define what the network is later on. And so he nicely said that in sort of that the total arborization of a neuron represents the graphic history of conflicts suffered during the developmental life. And I think that represents very much the absolute basis of what we are thinking about today in developmental care. How can we make this little neuron and its arborization actually get to the point that it has its optimal performance all throughout life? And people have looked at that evolutionary. So, for example, it's clear throughout the evolution, if we take the Neanderthal brain and we take the human brain, that one of the major part that is changed is the frontal lobe. So, when they reconstructed the skulls in evolution, they could see that really that there wasn't any big changes in the occipital or temporal lobe, but the main part was the frontal lobe that had grown tremendously between the Neanderthal and the human. Again, an indication that what socio-emotional functions which are assigned to the frontal lobe are probably the ones that over the years and in these growing and more complex societies have put more demand on the brain and therefore shaped the brain differently. So the early life events that we as caretakers of the mother, of the fetus, of the early newborn, or even of the extreme preterm, are actually working in this critical period of shaping the brain. So when we think in very simple terms of what is happening throughout gestation, then we know that the brain as an organ, of course, is formed very early on in the first weeks of pregnancy. That sort of the overall amount of neurons that in large we have for the brain are also formed in the first trimester. But then in the third trimester there is really this phase of organization of the final arborization and connections of these neuronal networks that is happening. There are intermediate zones in the brain like the radial glia and the sublight, which really guides the organization of the cortex, which then leads to this complex cortical folding that is really inherent to the mammalian brain. We know the rodent brain that we do all our basic science studies on is not a convoluted brain. So there is a lot of specifics to the human brain, which is laid down in the third trimester and therefore can be influenced with what is happening environmentally. The glial cell differentiation is happening in this third trimester, which assures myelination, which of course assures the transmission of information in our brain network. So prematurity, or anything that's happening during this transition of fetal to neonatal life, is most likely affecting all these basic neuroanatomical and neurodevelopmental processes. And as you see on this picture, where I was a couple of years younger, we started to try to get to these questions a bit better by using magnetic resonance imaging, which is a non-invasive technique that allows us to study, as you can see here in 3D, the forming of the human brain over the developmental period. So these are reconstructions of a human fetal brain and newborn brain, showing how this process of formation is happening. And we've, of course, studied these preterm infants, and we and others, many scientists in the world, and we've come up with quite distinct changes that are occurring by prematurity, and I can show them a little bit. As a summary, we've seen that throughout, let's see if this is actually working. No, doesn't. Okay. We'll do it the other way around. So from preterm to school age, you have differences in connectivity. You have smaller volumes when you compare preterm and full-term infants. You have changes in cell complexity. You have altered thickness in the cortex and surface. And you have altered resting state networks that Hugo and his team has worked on. So there are a lot of things that now, thanks to imaging, we can look at as the effects of an early alteration of development that lead to actually until school age changes. And I will try to show you some examples of that, particularly linked to where I think developmental care can make a difference. So when we look at these cortical developments over time, then we can see that cortical thickness doubles between newborn and school age. So there's massive processes occurring in this growth phase. And when we look at the thickness at school age, the one parameter, when you just lose the sort of general parameters of sickness in the newborn period and so on in the preterm population, it's actually the socioeconomic status of the parents that explains 25% of variability in the cortical thickness. So this very diffuse parameter of a genetic background, but of a stimulating probably environment has a biggest influence on the cortical thickness at 6 years of age. And when we look at how this cortex actually forms, we know that there are neurons migrating following the radial glial guides. They come from the ventricular zone. They form these neuronal columns that you see here. There is some tangential migration too for the inhibitory interneurons, but what you can see is that actually the cortical surface area is primarily driven by the number of radial columns that are present, whereas the thickness is really determined by the horizontal layers in the cortical columns. And these horizontal layers, if you remember, it's sort of an inward-outward organization. The first arriving neurons go to the basic layers of the cortex, and the last ones go to the surface of the cortex. So depending on when you disturb these processes, you can actually change the thickness of the cortex. People have started to look at this frontal brain that I've told you is probably the one that is most modulable by the environmental influences. And they've shown that the human orbitofrontal cortex, which links the reward mechanisms and the control of for example, reality filtering. What is it now? What is it tomorrow? What have I just seen? How do I integrate this? That this orbitofrontal cortex has a significant reduction in cell death in extremely preterm adolescents. It's also clear that there's a number of a sort of reduction of a specific mature type of cortical folding in the orbitofrontal cortex that persists until adolescent age. So these are clear new indications from neuroimaging that prematurity affects these networks. But then how can activity or developmental care shape the brain? So we looked also at specific whole brain connectivity. So looking at the entire network of fiber connections with a connectome approach, which is a complicated way of image analysis that we developed in a European consortium, which allows us to really illustrate all fiber connections within a brain. You see a bit of the cingulum, you see the uncinate. So you look at all the fibers, and when you do that and you do a connectome matrix, so every region of the brain is connected with another region of the brain, you can then look at the strengths of the connectivity by looking at the characterization of the microstructure of this connection between 2 points. And what we've then found was, when we compared the connectivity in extreme preterm versus control preterm infants, that these connections that you see here in red and in orange and in yellow were all weakened, whereas the blue one were strengthened connectivity. And the areas that happens, you can see, is predominantly the corticobasal ganglia thalamocortical loop and short corticals, particularly in the frontal area, but also parietal areas, whereas some of the connections within the temporal lobe were actually strengthened in the preterm infants. And when we correlated this to actual functional performance by using tests such as the KBC, simultaneous and sequential functioning, or social resolution tasks, and the SDQ for hyperactivity measurements and peer relationship, you can see that certain areas in the cognitive domain showed a high correlation of function and connectivity alterations in the frontal brain and in the orbitofrontal areas. So this is a strong indication that something that these extreme preterm infants go through leads to a altered connectivity in later life. And here are the areas that are particularly linked to social cognition and behavioral skills. And you can see that the orbitofrontal cortex connections appear prominently, whereas in the overall cognitive skill correlations, there are more different areas of the brain that are involved. But in summary of these network studies, we can say that the extreme preterm infant compared to a control preterm infant group of moderate gestational age has alterations in these corticobasal ganglia thalamocortical circuits in the prefrontal and in the corticolimbic area. So there we go and say, well, why is this happening? Where is this coming from? And of course, we know that the glucocorticoids as such, so stress hormones, are exactly modulating that axis and those corticolimbic structures. So in particular, the hippocampus, the amygdala, the nucleus accumbens, the prefrontal cortex and orbitofrontal cortex, and the cingulate gyrus are the ones that are controlling our emotional functioning and are affected by high levels of cortisol or by early separation. And this has been done in many studies by Michael Meany and others. In the NICU environment, of course, our baby is exposed and the parents are exposed to large cycles of stressors. And I just sort of illustrated that on this scheme. There is real stress, there is perceived stress in many situations. And so it's not too surprising to think that network is particularly modified by those environments that can come both antenatally, perinatally, and postnatally. So people have started to look at measuring these levels of stressors and brain structural outcome. This is some data from the US looking at the average stress scores, neonatal stress scores, and the bifrontal diameter of the brain or the microstructural maturity of fiber tracts. And you can see that the relative anisotropy is higher with lower stress scores. So how can we use this to think about developmental care or early interventions? Many years ago, when I was part of that consortium in Europe, Jacques Sison and Björn and the European Science Foundation Network said, we really need more research in this area. So have we done more research? Yes, we have done some research. This is data that I was doing when I was still in Boston, training with Heidi Als. And you can see that we tried to look at the brain networks for a group of babies cared for with NIDCAP interventions and standard care. And we can see that these connectivity that I was talking to you about was improved in the NIDCAP babies compared to the standard care babies. Nowadays, of course, we have a bit more sophisticated ways to look at that, but that was the initial days of DTI. And that also correlated, of course, with what you see here, the APIP with the attention score, and also with the later 9 months Bailey scores that confirmed this early intervention being beneficial both structurally as well as functionally. And Gloria and her team have looked at that even later at school age, and they can still find differences in the NIDCAP compared to the standard care, and especially in areas such as the RACE figure test, which is visual spatial working memory, which again locates the function directly to the prefrontal cortex. Just to tell you a little bit also that maybe prenatal interventions are also effective. This was a study where they looked at interventions to maternal caregiving during the pregnancy in high-risk pregnancies such as mothers with depression, and they did an intervention that could give high support or low support in the context of low or high depression severity, looking at adolescent age children from such pregnancies. And they could see that the hippocampal growth was influenced by the degree of the high support the mother would receive. So there are direct structural effects even in a prenatal intervention. When we looked at structural characterization of the brain, we could see that there were early structural biomarkers of language development in preterm infants. And so we said, well, this would be interesting. How much does a baby already actually perceive of a maternal language or voice perception or voice differentiation? Ghislaine, of course, had shown that at 3 months they already had a clear response with fMRI to language. And so we set out the study in saying, but if our preterm infants really hear mother's language for 10 weeks, then they should maybe differently process this compared to full-term babies that do hear their mother, but they hear it through a watery environment, so it cannot be the same kind of language. So we set up this study comparing preterm and full-term infants to mothers and stranger voices at 40 weeks. And we did this both with an EEG coherence setup as well as with an fMRI setup. And in short, just to show you the results, we can see that the full-term baby has a clearly different EEG map for the mother than the stranger voice, whereas the preterm, the two maps, they resemble each other. So the difference is actually bigger in the full term than in the preterm. And when we then compare forward versus backward language, then that is different. And then we can see that the preterm actually has a bigger difference between forward and backward than the full-term infant. And when we look at the fMRI results, we can see that again there is indeed a bigger difference in response from mother's voice to stranger's voice by the full term. But what the interpretation of these findings is that most likely the preterm baby, by its exposure to both the mother and stranger voice, actually processes both languages. And we have nice ways to see that this is actually true with activation in the superior temporal gyrus, which is the voice perception area in the adult brain also. And whereas in the forward versus backward, we have indications of activation in the more posterior part of the temporal lobe in the preterm infant, which would speak for a language processing. There's recent data that actually shows that this language exposure is beneficial. So the people looked at how many minutes the parents talked in the NICU, and the more the parents talked to the children, the better the Bailey language performance at 7 and 18 months were. We had looked at that early on. So if you look at, for example, the activity in the EEG in the first 72 hours of a baby's life, the higher the activity, so the more activity, the better is the subsequent brain growth and myelination. So clearly activity is boosting brain growth. And so the skin-skin contact, for example, which has been shown to increase the EEG activity, is one of these interventions that could improve brain growth by having more brain activity. And the recent study by Bill Fiver actually shows exactly that. This is actually affecting the most the frontal lobe EEG activity in these babies that were cared for in a family nurture intervention. And so we have neuroscientific evidence that these kind of interventions particularly address these prefrontal areas that seem to be impeded by the stressors of the preterm life. We've done some resting state studies as well to look at pain processing, and you see here data from full-term babies undergoing the Guthrie test, so the heel prick, and you see that this heel prick leaves a remembrance in the salience network of the resting state. So our babies do feel pain and process this pain post-pain situation. So how about music? And I give you just in my 2 last minutes a little bit of an idea how music could actually also be an intervention that we might be interested in. There's been quite a lot of studies already looking at physiological effects of music within the NICU. It's clearly something that falls into a critical period of development where the preterm infants are in the NICU. There is clear data from studies in brain injuries that music can help induce resilience and improve rehabilitation. And what you see here is data again from the Perani group showing that babies are able to differentiate the music that they have listened and the new music. And when we did this in our study, we could show that indeed there was, for a known music versus an unknown music, comparing preterm versus full-term infants, an activation of the ventromedial prefrontal cortex that we think is involved in emotion processing. I have to tell you a little bit that this music is specific, but given that I only have 1 minute, I can't have you listen to it. It's a specifically designed music. So I think I tried to give you a little hint on why interventions early on are important and why we have to monitor exactly what they're doing in a neuroscientific approach, and that we can use non-invasive tools to do that. From an economic efficiency point of view, clearly the earlier the intervention, the higher the economic return. And what I don't have time to do is to give you a little bit an outlook of what are the interventions beyond the newborn period, because they're as important than the newborn ones, but they will have to be better designed because they need to go to specific areas, and one specific area again in the frontal lobe would be the executive functions. There are preschool programs that improve cognitive control. These are data from Adele Diamond, clearly showing that if you do interventions that are in a social context, you can improve cognitive executive functions not directly related to your intervention in parallel. And that, I think, is an interesting concept. And we can use even their imaging to show rapid changes in structure by training, as these people have done in the hippocampus, showing changes in mean diffusivity after a video game play. So I think we are ready for neurofeedback, and we can have lots of things to work about. Then an emotionally nurturing environment producing more capable learners. And we need to concentrate on something that Heidi Als has given me the idea. She did a study in the 90s from cortex to classroom, and I think it's been my motto to continue that over the years, because we need to close the gap between what we know and what we do. And just to tell you that we will discuss that in the European Association for Developmental Care in October at the Scientific Symposium in Brescia, to which, of course, you're all invited. It will be going beyond the neonatal period into the pediatric side of developmental care and interventions. And I thank you very much for your attention.